Okay, so yeah, this is the biochemical analysis module. I can't remember which programs are doing it. I know it's the analytical bioscience, analytical chemistry as well. Structural biology. No. MRes students as well. Yeah. And yeah, that must be it. So I'm going to be doing most of the classes. I'm Philip Loudon, for those of you who didn't do the research skills module last year. And Professor Kostas Thalassinos, he's going to be coming over. He's a sort of joint appointment with UCL, and he will be teaching the mass spectrometry parts of the module. He runs the mass spectrometry facilities over at the UCL biochemistry department, and he's sort of international figure in mass spectrometry research. So just a quick overview of what we're actually going to be covering in this module. So it's the sort of general idea of practical techniques which you can use to analyze a whole range of different materials, but with a focus on biochemical samples. Also, I'll try and sort of put in a bit of sort of context later on about clinical applications, including some bit of sort of basic toxicology. And the kind of techniques we'll be covering, they're mostly based on the separation of molecules in a mixture into purer components. But that's things like liquid chromatography, especially high performance liquid chromatography, HPLC, but also sort of touch on some of the basics of ion exchange, size exclusion, chromatography, a little bit of gas chromatography. I will also be doing some electrophoresis. But warn you, this is kind of something I'm not that familiar with, but I've had to sort of take it on after one of my colleagues retired. So I will probably be keeping that sort of relatively simple. And yeah, as I mentioned, I'll sort of try and cover some of the sort of clinical applications, toxicology, and then there's going to be 5 sessions on mass spectrometry. So that's quite an important technique. And we're sort of bringing that into this module because it can be used with relatively small amounts of sample, and it can be linked up to some of the chromatography techniques as well. So some of you will be doing NMR spectroscopy and other sort of spectroscopic techniques in other modules that we're sort of focusing on the mass spectrometry here. And also it's got applications to protein analysis as well. So there's going to be, so we've got 16 lectures scheduled and 4 practical sessions. So the aim with the practicals is to sort of give you some sort of use of different types of chromatography instruments. So we've got HPLC, size exclusion, chromatography experiment, capillary electrophoresis instrument, and a gas chromatograph. And I think I sent out a message about this timetable, but I'll just comment on that now. I think I had someone commenting that there was a clash between practicals and lectures in some cases. I hope that's now been resolved. That should have, I sort of was in touch with one of the admin people on Friday about that, and maybe it's sort of taken a little while for that to get sort of altered. Has anyone sort of checked that this afternoon? And
on Thursday, 1 to 3. The third week is 1 to 3, or on Thursday, we'll have the first.
Yeah.
For lab session.
Yeah, so let me sort of see if I can remember what the clashes were. Yeah, so 18th, that should be a lab session. And that's going to be on the 6th floor, room 619. So next Thursday, lab. The following 2 Thursdays, the 25th and the 1st of February, that's going to be lectures. The 8th of February will be a lab. The other class.
Will there be an evening session for the lab? Will there be an evening session for the lab?
I'll come into that in a minute. So I'll have to sort of double check this and sort of put a note out on Moodle about the confirmed schedule.
It's 25th of January, then 2 sessions at the same time?
Yeah, that'll be a lecture. Yeah. So the following 2 weeks, Thursdays, next Thursday lab, the following Thursday lecture, and I'll confirm the others.
There's also one on the 15th of Feb, and
yeah, I'll deal with those. I'll sort of double check those and confirm that to you later. And there are lab sessions reserved on both afternoons and evenings on those days, and that's because there's too many of you to all sort of fit around one instrument at the same time. So you're going to have to sort of divide it into smaller groups to do the experiments. They don't take that long to set up. And a lot of the data collection is just sort of done automatically, so you don't have to watch the instrument all the time it takes to run. I'll need to get in touch with you to check what timing suits you best, and so I can organise you into smaller groups at a convenient time for you. But it's going to be basically between 1 o'clock and 9 o'clock. We'll be able to sort of fit you into the lab, and we might be able to fit in sort of 3 or 4 groups in the same day. And I also have some extra afternoon sessions where we've got the lab reserved in case we have to, sort of fit that in around the lecture sessions. Okay, so can I get on to? Oh, sorry, I forgot. I missed one thing when I was going through the content of the course. So what we're going to be starting with is not techniques for separating molecules, but for extracting them from the environment that they're sort of present in to begin with. So that's what I'm going to be doing today. And on Thursday, I'll be sort of introducing the chromatography and going through some sort of very basic theory of how we sort of quantify the separation of molecules in chromatography processes. Oh yeah, and the assessment, that's an important thing. So there will be an exam for this. So for some of you, this might be, I don't know, did you all do the exam? No, so not. So some of you haven't done the modules last term. I think the analytical bioscience maybe didn't do the module that had the exam last term, so it might be, for most of you, it's probably going to be your first exam here. As we can sort of get closer to that, I'll sort of say a bit more about the format of the exam, but it is intended to be an in-person exam. So you'll need to come in. It's booked for the evening of the 19th of March. And not sure of the timing yet, maybe sort of 2 hours. But as we, a couple of weeks' time, I'll be able to confirm a bit more detail about that. And the other half of the mark is going to be coursework, and it's going to be a mix of different things. So, there's going to be lab reports for the lab sessions that you do, but also some shorter problem-solving exercises to covering the lecture content.
Is it open book?
Sorry?
Is it open book?
Haven't figured out the details yet. I need to talk to my colleagues who organised the exam last term to sort of figure out how things went with that and decide on the best way to proceed. The thing is, the last few years we've been doing online exams, and they've not been entirely satisfactory. There's been some instances of people just sort of cheating and just using AI systems to write their essays for them. So we're sort of figuring out how to do in-person exams all over again. But sort of taking on sort of lessons from the last few years about how to make that more effective and a bit fairer. So I can't answer that yet, but I'll let you know sort of halfway through the term probably. There is the submission points on Moodle for the coursework should appear quite soon. I was going to, I think I might still, the first one was going to be next Monday as the deadline. I'll make that a very short exercise covering what we've been doing this evening's session. So it shouldn't take you too long to get through that, just something to get you started. So I think I'll stick with that deadline from Monday, next Monday for the first exercise, and I'll get that out to you tomorrow. It'll be things that I'll sort of fit in some sort of practice exercises this evening, the kind of thing that you'll be doing. Okay, so just a sort of general introduction to the. Sorry, I'm standing in the wrong place for this. Just going to get rid of this chair. Yeah, so we're mostly going to be covering techniques which are involved in separation of mixtures of molecules. Could be sort of small molecules in a sort of environmental sample or a biological sample, but we'll also touch on techniques that can be used to separate out proteins and nucleic acids as well, and also some of the, sort of the structural analysis that you can do using the mass spectrometry techniques. And there's all sorts of applications that you could choose, but just a few possible applications here. So, if you're synthesizing a drug molecule or something else that's going to be used in a biological system, you need to purify out the product from all of the reagents that you've used to make it. Or if you're doing protein expression, you need methods to purify the protein of interest out of that mixture. And that involves various chromatography methods. You can just use it even if your sample is almost pure. You might still need to do a bit of sort of cleanup to get rid of various sort of contaminants or other interfering compounds from the sample. And you can use some of these techniques just in a sort of very quick format, just to sort of kind of sort of take out one type of material, like ions, for example. Some of these techniques, especially what I'm going to be talking about this evening, can be used just as a way of concentrating a sample, and when you can combine some of the chromatography techniques with mass spectrometry. So at the same time as you're doing the purification, you can get some initial structural analysis of the molecules. So I'm not sure why the presentation keeps jumping, but I'll keep an eye on that. So I'm going to be covering things from a sort of fairly chemical viewpoint. So hopefully that's okay, fits in okay with most of your sort of backgrounds. For those of you who were sort of here last term, you'll have done the molecular basis of life module, and I think I remember that there was some sort of basic introduction to some of the chemical concepts there early on in that module. If you're only just sort of starting with us this term, you can have a look at this website here, which sort of gives some basic chemistry information. And it's just all to do with identifying different types of structural unit in organic compounds, like these, the ones that I've got listed here, various different types of structural unit that make up organic molecules and biological molecules. I'm not sure if those of you who just starting this term, you'll probably be doing the molecular basis of life module in the summer term. So you might be able to already access the Moodle materials for that. So you'll be able to have a look at some of those details and sort of have a look at the introductory sort of chemistry parts of that module. Let me know if you're having trouble accessing that. But I think I've had some people who just enrolled were sort of a bit confused by getting messages about the coursework on the molecular basis of life. So I assume that you can already see the Moodle pages for that module. Okay, so this is sort of still in a general introduction to the concepts we're thinking about here. So in order to sort of understand the techniques we're using to separate compounds, we need to think about the molecular properties of the molecules we're trying to separate out. And that's sort of physical and chemical properties, but also sometimes they are biological properties, mostly in terms of how they interact with other biological molecules. And so it's specifically things like the electrical charge on a molecule, an ion in that case, the polarity of the molecule, and that depends on particular types of functional group. Also, some techniques, the volatility matters, and that is linked to the boiling points of the molecules. The size of the molecule, so that can be the molecular weight, but also just the physical size that the molecule, and that's usually correlated with the molecular weight, but not entirely linear relationship between them. Also the shape of the molecule, and that's kind of tied up with the final sort of entry here. So a particular shape of a molecule can be recognized by a specific interaction with another molecule, a specific binding interaction, and that can depend on the three-dimensional shape of the molecule. And the last one, sometimes it can be like a chemical reaction occurring, which allows you to select a particular molecule, but it could also be a sort of non-covalent binding interaction that's occurring, like you get between an antibody and what's the word? What do antibodies recognise? Yes, thank you. And this is just a very small sort of sample of the kind of applications that are possible for some of these techniques. So I'll be talking about solvent extraction and solid phase extraction today, which those of you who did the practical classes last term, you've done these techniques already. And they're covered in the research skills module, so the rest of you will be doing that next autumn term. So for solvent extraction, that's a very old technique, but it's still used for getting material out of, usually from an aqueous solution, and then concentrating it into and sort of separating it out from more water-soluble materials. Solid phase extraction, that's frequently used for just a quick sort of cleanup of a sample, but it can, and concentration of a sample, but it can also be used as in my experience, it can be used to do sort of a little bit of a sort of crude chromatography effect as well. And I didn't really emphasize this in the lab classes, but when we did the DNA isolation in the lab classes last term, that was also a form of solid phase extraction, because we were putting blood sample through a membrane, and the DNA molecules were getting bound to that membrane. So it's kind of the same principle as solid phase extraction. So gas chromatography, that's used to analyze volatile materials because they have to get into the gas phase. So that could be things that if you're analyzing things like petroleum or sort of similar kind of hydrocarbon materials, or other volatile things like fragrance molecules, that whole wide range of other things. Even archaeological samples can be analysed by GC in some cases. Liquid chromatography, it's a very common technique. So there's all sorts of applications, but it's used in organic synthesis. You can use it to purify the final product of a synthesis, but also widely used for the analysis of drugs, both just therapeutic drugs, but also drugs of abuse or sports doping, that sort of thing, as well as drug metabolites. Thin layer chromatography, I might not get into the theory of that so much. Again, it's something that comes up in the lab classes in the autumn term. So you can use that to check whether, if you're doing a chemical synthesis, you can check whether it's worked or not, or you can sort of look to see how many compounds you've got in a biological mixture. Then ion exchange chromatography, that's widely used for protein purification because different proteins have different numbers of positive and negative charges on them. But it can also be used to take out simpler ions, so it can be used in water purification processes, either drinking water or for deionized water for lab applications. Size exclusion chromatography, again, used in protein purification because it's based on molecular weight of the molecules, and proteins have a wide range of different sizes and molecular masses. But it can also be used for synthetic polymers as well, to make sure that if you're doing a polymer synthesis, that you can allows you to restrict the range of the lengths of the polymer molecules. And finally, electrophoresis, which can be used for purifying both DNA and protein based on the different charges. You can use it to, it's mostly used for separating out molecules that are charged, and it will then be based on the size, the molecular mass of the molecule, and the size of the molecule. So I'm still getting to grips with this before I do the electrophoresis lectures, but you can also modify it to separate out neutral molecules as well, with sort of various modifications to the underlying electrophoresis technique. Okay, so before we get into discussing the actual techniques in practical terms, we do need to just sort of do a little bit of groundwork to think about the kind of physical processes involved here. So, in order to use these separation techniques, we are relying on the fact that different molecules have to interact with the experimental materials in different ways, and this is all based on differences in non-covalent interactions, which are sort of intermolecular interactions which don't involve forming chemical bonds. So, these, I'm going to go through these sort of various, essential intermolecular interactions. But these will influence the polarity of molecules, which influences how strongly they bind together. It also affects the volatility of compounds, so how easy it is for them to get vaporized, and and that's sort of linked up to boiling points. And yeah, sort of commented on the kind of interactions that we might be thinking about, or the kind of molecular properties we might be thinking about. So we're going to consider the interaction between ions, so that's charge-charge interactions. Also, not just fully ionized molecules, but also partial charges that develop in molecules with certain kinds of chemical bonds. And that, that's where you generate the dipole, so-called dipole moments in the molecules. And also, you should have all heard of hydrogen bonding, so I'll discuss that a little bit. That's specifically where you have polarized bonds to hydrogen, usually oxygen, nitrogen, or sulfur attached to hydrogen. But also discuss interactions that can occur even with non-polar molecules. Okay, so first of all, interactions between ions. So ionic bonding can be referred to as. Just a quick reminder, talking about cations, which are positively charged, anions, which are negatively charged, and going back to basic physical chemistry, the interaction of ions is described by the Coulomb's law, which I'll quantify on the next slide. So, ions with the same charge are going to push each other away from each other. Ions with opposite charges will be sort of drawn together. And things like sodium plus, chlorine minus. And it's a sort of fundamental principle for how sort of different salt materials form, like sodium chloride. So I'll quantify this in the next slide, but it depends on the size of the charge. So if it's a fully formed ion, that could be varying different whole numbers of charges. Most commonly, sort of 1 or 2 charges on each ion, but in some cases it could be higher. But it's also going to depend on this distance between the ions. So we'll see the distance relationship for this the strength of the interaction in a minute. And it also depends on what's around the ions, the solvent that they're dissolved in, or sort of more generally their sort of local environment. Not going to sort of really get into this too much, but that's quite an important thing when you're thinking about the formation of protein three-dimensional structure, because the ionic interaction, it depends on which part of the protein structure the ions are found in, and it can be sort of stronger or weaker depending on what other sort of protein structure is around it. So here's the equation that describes the interaction of ions that comes out of this Coulomb's law. The energy of the interaction, so this is going to be, when the ions come together, usually we're thinking about ions of opposite charges, they're sort of coming together, and that's lowering the energy of the system. And we're thinking about the strength of, the amount of energy that's released when the ions come together. So it depends on the charge on each of those ions. That's the Q terms in the equation. It's inversely proportional to the distance between the ions. So the closer they are, the stronger the interaction. And it's also dependent on this value here, it's epsilon, and that's described as the relative permittivity of the environment that the ions are in. And it's also referred to as the dielectric constant. And it's sort of, so I keep forgetting words. It's normalized to vacuum So if there's nothing else around, it's sort of set to 1, then everything else is sort of related to that. The basic, 1's the lowest, and any other material is going to be higher than that. For non-polar environments, it's relatively low. So for hexane, the hydrocarbon solvent, it's 1.9. For more polar liquids, it's much higher. So ethanol, 24, and water, which is even more polar, is 78. So, what that means is, think about this. The strongest interaction is when there's nothing else around the ions and they're just in a vacuum. But if it's in a non-polar environment like a hydrocarbon solvent, then it's still a very strong interaction. But it gets kind of weakened if the ions are surrounded by polar molecules like water molecules. Then there's another kind of sort of charge-based interaction that we need to consider. And this is where we've got neutral molecules which have still got some polar character to them. And this is because, any bond between the atoms of two different elements is not going to be, it's not completely symmetrical, and you will get a slight distortion of the distribution of the electrons within those chemical bonds, and some atoms of some elements will sort of draw the electrons closer towards them compared to the other elements. So what it means is that, certain types of bond will have quite a significant shift of the electrons from one atom to the other. And that means that one atom has a greater amount of electron around it, and the other atom will have its positively charged nucleus more exposed to the outside world. And that means that we end up with this, described as partial charges at each atom, which can cause interactions with other molecules which also have these partial charges. And it's the same kind of principles involved. So opposite partial charges will be able to sort of attract each other and draw the molecules together. And we can sort of quantify this in terms of dipole moments. This is usually applied to the whole molecule. I mean, I guess you could also sort of think about it for individual bonds as well, but it's usually describing the whole molecule, where you sort of add up all of the effects of all of the different chemical bonds. And it's, what you have to do is to sort of calculate what the partial charges are, and how far apart, if you're thinking about a chemical bond, the R is going to be the bond length, and Q is going to be the how much the charge has been separated across that chemical bond. And it can be given units in, this is a coulomb meter, so coulomb is a measure of charge, meters just distance, but it's also referred to as Debye units. That's just named after some physicist from the 19th century, I guess. We're not really going to get into sort of quantifying this too much, though. It is worth saying a little bit about where this comes from in terms of the properties of different atoms. So, if you've done any kind of basic chemistry study, you should have come across the concept of electronegativity. I don't really have time to sort of go into all the full definitions of this at this point. But electronegativity, it comes out of the atomic structure of each different type of atom, the precise sort of arrangement of the protons in the nucleus and the electrons that surround the nucleus. And it's an empirical term, so it comes out of looking at the properties of lots of different molecules and looking for patterns in how different atoms behave within those molecules. And so you can come up with these numbers in various different ways of processing all of the chemical data that's available. And there's different ways of calculating these numbers, but the trends are always the same. So fluorine usually comes out as the highest electronegativity. So if you've got a fluorine atom in a molecule, it's going to pull electrons away from any atom that it's bonded to. So the bonds are always going to be polarized, with the fluorine carrying a sort of noticeable partial negative charge. So it's got more electrons close to it. Oxygen is relatively high as well, and that's significant because we're going to be thinking about the properties of things like water, methanol, alcohol molecules. The other halogens like chlorine are also relatively high. Nitrogen fairly high, but it's getting down a little bit further down from oxygen. Sulfur a little bit less. Carbon, because we're talking about organic and biological molecules, they've got lots of carbon atoms in them. So that's usually what we're sort of thinking about is what's attached to the carbon atoms. So that's relatively low, and hydrogen is lower still. And what you're doing, if you're thinking about, how polarized is a particular chemical bond, you sort of think about what atoms are being joined together, which elements are being bonded together, and you sort of look at the difference in these electronegativity values. So, the most extreme example, actually things like sodium, potassium, they'd be even lower. So they've got such low electronegativity that they're easily ionized, so they completely lose the electron. But if we're thinking about covalently bonded molecules where you've got a full chemical bond between them, the most, one of the biggest differences is going to be between hydrogen and fluorine. So that's a large difference in electronegativity, tells you it's a very highly polarized bond. Okay, so just trying to remind myself what I was going to say here. So here's some examples of molecules which have got these polarized bonds. So hydrochloric acid in its molecular state, got hydrogen bonded to chlorine. Quite a big difference in electronegativity there, so it's going to be a very polarized bond. The chlorine is pulling electrons away from the hydrogen, so it's picking up this partial negative charge, and the hydrogen is left with its nucleus more exposed to the outside world, so it's got a partial positive charge. And the partial charges are represented with this delta symbol, commonly. With the next molecule, methanol or formaldehyde, that's got a carbon-oxygen bond, but it's a double bond, and that gives you an even sort of greater, that enhances the polarization of the bond. The oxygen is more electronegative than carbon, so it's pulling electrons away from the carbon, so the oxygen ends up with a greater amount of electron density around it, so it has the partial negative charge, and the carbon has had electrons pulled away from it, so it's got the partial positive charge. The final example is an amine, ethyl amine, where we've got NH bonds. Nitrogen is more electronegative than hydrogen, so it has the partial negative charge, and the hydrogen is with the partial positive charge. So the idea here is that if we sort of think about what the effect this is going to have on extraction processes, separation processes, These polarized bonds can interact with polarized bonds on other molecules via these electrostatic type interactions. And we need to sort of be able to sort of think about how strong an interaction this might be, depending on how polarized the bonds are, and how that will affect the strength of the interaction between different molecules. So, this is just an example of showing you hydrochloric acid molecules sort of coming together, and it's based on the attraction between these partial charges on the hydrogen and the chlorine atoms. But an example of interaction between these dipolar bonds and ions. So this could be I think it's the sodium or potassium ion here, and these are the nucleobases that you'd find in DNA. This is an observed structural feature at the end of chromosomes. I think it's called a quadruplex structure, where you get 4 DNA strands sort of wrapped around metal ions. So the R groups, that's the DNA chain coming over here. And so we've got these carbon-oxygen double bonds, so they have this quite strong dipole where the oxygen atom has a partial negative charge, and that can interact with the positively charged metal ion. And it's quite a good example to link to the next section, because it involves these other interactions between the so-called bases wrapped around the metal ion, indicated with these dashed lines, where we've got bonds to hydrogen, between hydrogen and nitrogen, interacting with oxygen or nitrogen atoms, and that's the well-known hydrogen bond that's being illustrated there. So, I'm going to take a bit of time to discuss hydrogen bonds here. Very important type of interaction. Obviously, we've kind of hinted at that. It's the absolutely fundamental for the way that biological molecules work. It's what links the DNA bases together, which gives us rise to the genetic code. It's the basis of how protein structure forms through the formation of things like alpha helices, beta sheets. That's all based on hydrogen bonding. And it's also kind of important for the fact, the properties of water. So it's hydrogen bonding that's mostly holding water molecules together. So what this involves is effectively a hydrogen atom being shared between different atoms to some extent. It requires a bond between the hydrogen and a very electronegative element, a relatively electronegative element like oxygen, nitrogen, or sulfur, are the most important ones usually. But it has to interact with another atom which has a lone pair of electrons available to interact with the hydrogen. So it's got a pair of electrons sort of sitting on the outside of the atom, which are sort of giving you this sort of concentrated zone of electron density with a partial negative charge. So the hydrogen bonding, it involves some kind of charge interactions, a sort of dipolar interaction involving these partial negative and positive charges. But it's got this sort of very specific character compared to other charge interactions, where you can actually get the beginnings of covalent bond formation occurring between the hydrogen and the other atom with the lone pair. And that leads on to a kind of a unique, I don't know if it's unique, but maybe almost unique feature of the hydrogen bonding. So not only is it quite strong, because it involves the interactions between these opposite charges, but it's directional. So it works best at particular three-dimensional arrangements of the atoms. I've got this sort of bond strength there. I can't remember how strong a covalent bond is, though, so I can't really comment on that. So, just a little bit more detail about them, and some of the nomenclature involved here. So the molecule, or the part of the molecule which has the bond to hydrogen, OH, NH, or SH bond, that's referred to as the donor, hydrogen bond donor. So the hydrogen is attached to the donor atom. The acceptor atom is the one which has got the lone pair, which is interacting with that the bond to hydrogen. And with the lone pair of electrons. This is the sort of typical kind of distance that you would get between the atoms involved. And just sort of looking ahead to when we sort of start to think about the properties of different solvents, I'll be sort of mentioning so-called aprotic solvents, and that's where you don't have this kind of bond to hydrogen, which can take part in hydrogen bonding. So it's just a look ahead to what's coming up. Just some more examples of the kind of combinations that you might see in biological systems where hydrogen bonding occurs between amines and water and carbon-oxygen double bonds. So you see water's coming in a lot. It can take part in all sorts of different hydrogen bonding interactions with itself, between the OH bond and the oxygen atom on another molecule. And top right, that's very important for protein structure, because that would be an example of the hydrogen bond that holds the two of the peptide bonds together. So we've got NH, which is part of the peptide linkage between amino acids, and that can form a hydrogen bond to the carbon-oxygen double bond of a peptide linkage elsewhere in the protein chain. And here's just a little bit more about the directionality of these hydrogen bonds. What's involved here is, you need to fully explain this, you need to sort of start thinking about it in terms of atomic orbitals, molecular orbitals, a sort of quantum mechanical description of chemical bonds. And this, you get this overlap of these orbitals, which are sort of involved in forming bonds in molecules. And if you sort of model this theoretically, what you find is that it depends on the precise positioning of the molecules, where you get the optimum interaction of these orbitals. And it's strongest when you get this sort of completely linear arrangement of these atoms. So if you think about the angle between the XH bond and the HY hydrogen bond, if they're completely linear, that angle is 180 degrees. And what this graph is showing you is the reduction in energy when the molecules come together to form the hydrogen bond. It's greatest at that point. There's a little bit of flexibility though, so it can sort of twist around a little bit either side, but not all the way round. So once you twist it round to a certain point, then the strength of the bond decreases, and you get a sort of much lower decrease in energy as the molecules come together. Okay, so trying to make things a little bit more interactive at this point. So we've got some more examples of molecules here. And I just want you to think about which of these molecules could act as an acceptor for a hydrogen bond. So they need to have an atom which has lone pairs of electrons on it. So that's pairs of electrons which are not involved in bonding, but they're sort of, exposed at the surface of the atom. So think about that first. So I'll give you just a minute or two to have a think about that for each of those 4 molecules. So I'm not going to ask anyone to kind of explain to me what you're thinking. I'm just going to ask you to sort of raise your hand for each molecule in turn. So think about the top left molecule. So could that be a hydrogen bond acceptor? Just sort of raise your hand if you think it can. Nobody? Okay. It is. So that nitrogen atom, that's available. Nitrogen atoms have lone pairs of electrons on them. And that nitrogen atom there, that could take part in hydrogen bonding as an acceptor. Okay, so what about the top right? Could that be a hydrogen bond acceptor? Just raise your hand if you think so. Yeah? Yeah, so the oxygen atoms also have lone pairs, so they can take part in hydrogen bonding. So bottom left. Yeah, so we've got a couple of people shaking their heads. Yeah, that's right. Although it's got a nitrogen atom, the nitrogen atom has been protonated, so it's had a H plus has been attached to the lone pair, and that is now no longer accessible to anything else. And bottom right, hydrogen bond acceptor. Yeah. Again, because of the oxygen atoms, they're sort of poking out and available. So, I won't sort of go through all of every single hydrogen atom in detail. Just first of all, for each molecule, could it be a hydrogen bond donor at all? So have a think about that for a minute, and then I'll sort of go through each one in turn. So don't worry about specific hydrogen atoms. Just is there anywhere in the molecule that can act as a hydrogen bond donor?
On the second one, two oxygen. Not so it is the oxygen in the middle. This one, like one of the hydrogens from there, that carbon can use the hydrogen and then double bond to it's about vaguely remember something. My understanding.
Yeah, it need might need other sort of reagents to think about the structure as it is. Okay, so that's the top left. Could that be a hydrogen bond donor? No. It doesn't have any bonds between hydrogen and electronegative atoms like oxygen, nitrogen, or sulfur. So I'm sure we've got all of the hydrogen atoms drawn in, but those ones they're all to carbon, and there's not enough of polarization of those bonds. So top right, hydrogen bond donor. No. Because again, the only bonds to hydrogen are from carbon atoms. So this one's near an oxygen, but it's not near enough. And with a strong base, you could remove that hydrogen, but it's not sort of coming out very easily. It's a little bit more polarized than the other ones, but not very. Okay, so bottom left, could that be a hydrogen bond donor? Yeah, I've got some people nodding their heads. So yes, we've got 3 hydrogens attached to a nitrogen atom. Each one of those NH bonds is going to be very polarized, so those hydrogen atoms can interact. And it's got positive charge on it already, so that kind of enhances the effect. But not the CH bonds. And finally, the bottom right molecule. Yeah, got some people nodding heads. Yes, it's got NH bonds. They're polarized away from the hydrogen, so they can hydrogen bond. Just the NH bonds, not the CH bonds. Go ahead.
I have one question actually. This same rule goes on the intramolecular hydrogen bond as well?
Yeah. Because hydrogen bond. Well, we're usually thinking about it in terms of different molecules coming together. But if it's a big molecule like a protein, then it's effectively intramolecular. But you can think about it as different parts of the molecule coming together. So it's usually sort of. So with alpha helices, it's quite close. But mostly it's bits of the protein that are far away from each other in the sequence and come together by hydrogen bonding. And the final interaction I'll be discussing. Hang on, let me just check the time. Okay, if I just sort of finish this off, be another sort of 5 or 10 minutes, just to sort of finish off this theme, and then we'll have a break. Even where you don't have any significant development of charge in a molecule due to an ion or a polarized bond, it's still possible for molecules to sort of have some weak interactions. Otherwise, things like petrol wouldn't be a liquid if all the hydrogen carbon molecules couldn't sort of stick together somehow. But where you've got, generally with non-polar molecules, things like hydrocarbons, they don't have very strong binding interactions, pulling them together. But it depends on the size of the molecule as well. So the bigger the molecule, the more opportunity there is for them to stick together. And even things like the noble gases like argon, neon, if they bring the temperature low enough, they can sort of come together to some extent and liquefy. And what is happening here is that atoms and molecules, even if they don't have these sort of permanent distortions in the electron distribution, the electrons are not totally fixed. So there's sort of constant fluctuations in exactly where the electrons are in a molecule or an atom. And you can get these sort of very short-lived, transient formation of sort of charge separations, dipoles within a molecule, even if it's overall sort of very non-polar. And if you've got two molecules close together, if you get this sort of transient dipole forming in one molecule, that can induce a similar dipole in the neighbouring molecule, because if you've got a little bit of negative charge sort of moves out towards the other molecule, that can repel electrons in the other molecule, and you end up with this sort of very small sort of positive negative dipole on both molecules, and it sort of pulls them together slightly. And there's various names for this type of thing, and there may be some sort of slight differences in the definitions of these, but you might see them described as dispersion interactions, London interactions, quite commonly as van der Waals interactions as well. They're very weak, and they only work at very close range. So if you look at how the strength of the interaction changes as you separate the molecules, it varies by 1 over the distance to the power of 6. So it only works if they're very close together. That has implications for protein structure. So if you're thinking about how sort of the hydrophobic bits of a protein stick together, you have to have this sort of very close matching up of the surfaces of the protein that are coming together by this type of interaction. Very complementary shapes. A consequence of this, just in terms of small molecules, is that if you have a non-polar molecule which doesn't have this a significant permanent dipole, it's only going to be, the molecules can only be held together by weak forces, so they will sort of have very low boiling points. So if you take these, different molecules, each with it's got one carbon and different amounts of polarization. Methane, it's completely symmetric. Well, not completely, but it's got a high degree of symmetry, so it doesn't have any kind of overall dipole in the molecule. If you look at the whole shape of the molecule, so it ends up having a very low boiling point. Methanol, or formaldehyde more commonly, it's got carbon-oxygen double bond. That gives it a permanent dipole overall, but it doesn't have any hydrogen bond donor groups in it, so it's got a relatively low boiling point. If you add a couple of hydrogens to it to get methanol, we've now put in an OH bond, so that can take part in hydrogen bonding, so that gives a greater strength of these intermolecular interactions between methanol molecules, and so it's now got a much higher boiling point. Okay, so I think that's a suitable place to take a break. So it is now, okay, 20 past 7, not 20 past 8. So sort of start again about 7:40, is that okay? Or do you want to sort of start a bit earlier?
Let's start earlier,
so we can finish.
Yeah, so 15-minute break enough? Yeah. So I'll start 7:35. Just in case there's a queue at the coffee bar. Okay, so we're getting on to thinking about extraction procedures, and this is mostly going to involve the use of different solvents, and it's also relevant to some of the chromatography techniques as well. So we need to be able to think about the properties of different solvents and how that affects the way that they interact with the molecules that you're trying to dissolve. And it's mostly based on polarity of the solvents, and this, governs how the types of molecules that they can dissolve. So you're sort of well aware of this sort of thing that water will dissolve similar types of compounds, polar molecules. Non-polar solvents will dissolve non-polar materials. Polarity, you'll see this sort of discussed a lot in sort of all sorts of literature, but it doesn't have a very specific definition, and it depends on the sort of varying types of intermolecular interaction that I've been discussing. All of these different examples. Sorry, wrong way. But you can sort of take different types of molecule which act as solvents and roughly group them into different types. So you can get extremely polar molecules, which are not very commonly used as solvents, but which have, things like strong acids, which actually sort of dissociate and ionize themselves under the right conditions. So things like nitric acid, sulfuric acid, they can act as solvents, but they're obviously not very nice things to work with. More commonly, it'd be highly polar molecules like water, methanol, ethanol, molecules which have OH bonds, which can form strong hydrogen bonds to hydrogen bond acceptors, but also accept hydrogen bonds from OH, NH bonds on the molecules that they're dissolving. Then fairly polar molecules. Now, if you note, these have actually got, some of these dipole moment measurements are actually bigger than for the very polar, the highly polar solvents. But none of these molecules has any OH or NH bonds, so they can't form hydrogen bonds as a hydrogen bond donor. So these are described as aprotic, but dipolar. And it turns out that they are quite good at dissolving polar molecules, but not quite as good as water, methanol, ethanol, etc. Probably worth, I won't go through it here, but it'd be worth you looking up the structures of these molecules so you get an idea of where the polarity is coming from, what kind of bonds they contain. Then there's molecules which are slightly polar, so these are weakly polar solvents. They're also aprotic, so no OH-NH bonds, but they've got relatively small dipole moments, so they're not as polar as the previous lot. Diethyl ethers quite common, despite the fact that both of these solvents, they're quite commonly used, but they're quite unpleasant things to work with. And then we've got non-polar solvents, things like hydrocarbons like pentene, hexane, toluene, benzene also, carbon tetrachloride, another nasty compound. Benzene's quite nasty as well. But see, these ones, these are almost completely non-polar. They've actually got zero dipole moment. Got quite a lot of symmetry in their structure. So now we need to think about mixing solvents together. So this is relevant to some of the extraction procedures we're going to be discussing. I'm not going to get into heavy thermodynamics, but this is one of the main outcomes from thermodynamic theory. This idea of the Gibbs free energy, so that in a sort of complex system, things are going to sort of arrange themselves to reduce that gives free energy value and sort of put energy out. But it's combined from the enthalpy. So that's usually to do with forming chemical bonds. That includes the non-covalent bonding that I've been discussing. So the stronger the non-covalent interactions we have, the higher the enthalpy will be, and the greater the stability of the system. But it also depends on the entropy, which is temperature dependent as well. So that's a measure of the molecular disorder that we have in the system. So, very roughly, if you think about something going from solid to liquid to gas, it's got the greatest order in the solid state with all of the molecules sort of fitting together in sort of specific arrangements. The entropy is greater in the liquid state, where the molecules can sort of move around each other, and even greater in the gas state, where they're sort of separated from each other and they're sort of free to move around much more rapidly without any sort of constraints on their orientation. So, because you've got the negative term there, as the entropy increases, so disorder increases, that stabilizes the system, so it gives you an overall reduction in energy. Effectively, it takes an input of energy to organize molecules into a sort of ordered state. And this is relevant for if we're sort of mixing different types of solvents together. So if we take just water and a hydrocarbon of any non-polar solvent like pentane, they don't mix together. They will just sort of give you these sort of two separate layers. And this comes about from what's called the hydrophobic effect, which is quite a difficult thing to explain, but I'll do my best. And it's still a bit of sort of discussion about exactly why it works. But yes, so basically it has lots of consequences. So as well as just sort of mixing different liquids together, whether they dissolve in each other or not, it governs the way that lipids behave in biological systems. So the formation of cell membranes depends on the hydrophobic effect. The folding of proteins into their three-dimensional structure, that is also affected by this hydrophobic effect. And I'm just going to think about it in this, discuss it in these sort of simple terms of water and hydrocarbon solvent. So it's all to do with, mostly to do with the thinking about the entropy of the water molecules particularly. So there's lots of non-covalent interactions between water molecules. So there's high enthalpy factor when water molecules come together. But if you put in some pentane, you can still imagine that the water molecules, they can still sort of go around and make hydrogen bonds with each other. But when they are next to some pentane molecules, where the water molecules face the pentane, they can't form hydrogen bonds because hydrocarbons don't have hydrogen bond acceptor groups. So there's a reduction in the enthalpy factor there. So we're not getting so much bonding, non-covalent bonding occurring. But there's also, if a water molecule is sort of close to a pentane molecule, it also constrains how it can form hydrogen bonds with other water molecules. So that leads to a decrease in its entropy as well. So it's overall unfavourable for the pentane to be mixed in with the water. And I'm sort of trying to illustrate that issue here. So if you imagine sort of having some, little sort of cluster of pentane molecules here, hydrophobic molecules.
Yeah, in a biological context, where is pentane related to?
It's not that common in biological systems. Just using it as a very simple molecular example, but also relevant to the extraction techniques that we'll be discussing. So, try and imagine, you've got this cluster of hydrocarbons here. Could be pentane, could be a bit of a cell membrane. You have some lipids, same principles. Away from that, so just in the sort of bulk liquid state of water, you've got these water molecules, they're forming hydrogen bonds with each other, but they can switch around. They can sort of turn around and form hydrogen bonds to other water molecules. So there's a bit of disorder there. They can just, move around and always still be able to find the hydrogen bonding partner. So we're not losing the enthalpy factor by the water molecules moving around, and turning around. But when they get close to this non-polar blob here at the top, they can't form hydrogen bonds to the hydrocarbon up here. They still need to try to find hydrogen bonds to neighbouring water molecules, but they are more sort of constrained into how they orientate themselves in order to form these hydrogen bonds. So effectively, as the water molecules sort of get closer to the hydrocarbon, they're having to adopt more sort of rigid positions in space to be able to maintain the hydrogen bonding interactions. So it's kind of, they're almost getting close to the solid state. They're sort of more fixed in their orientations, almost like they're sort of starting to freeze around the hydrocarbon region. So what the system will sort of do is that, the hydrocarbons will sort of cluster together as much as possible, so we minimise the amount of the surface area between the hydrocarbon and the water molecules. And that means that we get as much disorder in the water as possible, giving us a high entropy factor. Okay. Right, so now we're finally getting on to some of the practical techniques. We'll start off with solid phase extraction. There's not a lot of sort of theoretical discussion here, so sort of relatively straightforward. But what you have is a solid material which has some kind of absorbent properties, and there's sort of different types of material can be used to absorb different types of molecule. And what's happening here, you're going to have non-covalent interactions between the sample molecules that we're trying to isolate and the surface of this solid material. It's most commonly used with liquid samples. So if you did the lab classes last term, you've been doing that, antibiotic solutions, we put those through solid phase extraction cartridges, the antibiotic molecules bound to the solid phase material, and then we took them off with a different kind of solvent to dissolve the antibiotic molecules off of the solid phase extraction material. And it's all, I've sort of mentioned that it's the principle behind a lot of DNA extraction techniques, where the DNA binds to a particular sort of polymer material under certain solution conditions, and then we change the environment so we can dissolve the DNA off of the membrane. And there's a sort of wide range of different type of materials. So we can have polar materials, and this can just be sort of minerals like silica, silicon oxide, and that can bind polar molecules, and that, I'm sure you would use hexane so much. Non-polar materials, we can sort of take the silica and add hydrocarbon chains to it to turn it into a non-polar surface. We can have materials with ionized groups on them, which can bind to ions from your sample solution. So it could be negatively charged ions like CO2 minus, carboxylate groups, which can bind to metal ions like sodium. or positively charged groups. So this is a bit tricky to see what's going on here, but it's an amine group which can pick up protons which have positive charges on them, and so it can bind to anions like chloride. You can have more specific interactions. So I'm not sure that's quite a good example of the DNA, but you can have, if you've done any protein purification using affinity tags, where you have a very specific binding group which will bind to a specific sequence on your protein. That's kind of effectively a solid phase extraction method, where you've got your sort of column which has the complementary binding group, and only the protein which has that particular sequence will bind to the column. Everything else just washes through. And the sort of general idea is that there's two main applications. Go ahead.
The previous things is used in liquid chromatography as well, like polar and reverse phase.
Yeah, we'll get to that shortly. So in a later session. So you can use this in a way of just sort of doing a quick cleanup, where you choose the sorbent material, the solid phase extraction material that doesn't bind to the thing that you're interested in, but does bind to other things. So the species of interest that you're sort of trying to clean up just passes through the material, sometimes referred to as a matrix. So this is often used with like an ion exchange or ionic material, where you're sort of trying to deionize a sample, and so you can sort of, take out some of the ions from the mixture. But you can also, what we did in the lab classes was the other application, where you're trying to bind the molecules of interest onto the cartridge and let other things wash through. And then you use a different solvent, which weakens the interactions between the sample molecules and the binding matrix. And then that comes up, you wash off your sample molecules in a second step. And you can use that as a way of concentrating your sample, because you put in a large volume to start with to load your sample, and then you wash it off, you elute it in a lower volume.
So previously we used the second technique, right?
Yes, that's right.
So there are obvious practical benefits to that. What are the other benefits? Solid phase.
Yeah, I'm not sure I follow. It depends on
in what context would it be functional?
I mean, I use it a lot to isolate natural products from bacterial cultures, for example. It's a bit more expensive than solvent extraction, but it's a bit less labour intensive, a bit less environmentally harmful sometimes, depending on what solvent you're using. You can use lower volumes of solvent.
And the quality of the product according to the extraction.
It's hard to generalise. Depends on the precise application, really. And just a quick run through some of my favourite materials. So this is the ones we used in the class, or similar to that. Companies will sort of manufacture specific polymer materials to optimize the behavior. So there's this stuff called HLB, which I think it stands for hydrophilic lipophilic balance material. And it's this sort of complex polymer. It's got non-polar groups, these benzene rings, but also polar groups, these amide bonds. So it allows you to bind a wide range of materials at the same time, with different kinds of interactions occurring. And it's been modified to introduce charged groups, so you can have these positively charged ammonium groups. which can bind also to negatively charged ions in your sample. And instead of that, you can add negatively charged groups, SO3 minus groups, sulfonic acid groups, which can bind to positively charged ions in the sample. So say just a little bit about solid phase extraction. You'd use that if you have a liquid sample that you're trying to extract something out of. But you may have a solid material that you need to extract, like this is used a lot in plant natural products, where you have some plant material and you're trying to dissolve out interesting natural products from the plant, or I analyze soil samples for sort of different sort of potential environmental pollutants. So it sounds quite simple. You just take your solid sample and you just put some solvent. You grind it up maybe to sort of give sort of smaller particles, bigger surface area, and then just mix it with some solvent. And that's a very simple way of doing things, but it can require quite a lot of solvent to dissolve enough of the material of interest, and then you have to spend time and energy evaporating the solvent afterwards. So there have been ways developed to optimize this, to use less solvent, but it requires a specific apparatus to do this. I'm not going to go through the theoretical basis for this, but it's to do with the fact that usually the material you're trying to extract will be less volatile than the solvent that you're using. So if you mix them, if you have a solution of your sample in the solvent, you can sort of keep on, you can boil the solvent, and most of your sample molecules will sort of still be in the liquid phase because they're less volatile. They've got a lower vapor pressure, so we have quantifying volatility. And if you use this kind of apparatus, what you can have is a solid sample where you're continuously passing the solvent through it, and you're effectively recycling the solvent during the process using a much lower volume of the solvent. It's better as a sock slit apparatus. I'll sort of explain the design of it on the next slide. But there's a video. I'll put a link to this on Moodle as well, so you can sort of see it in action. But I'll sort of do my best to explain it here. So this is your solid sample, so it could be sort of some plant material, and you put it into this. This is a kind of like permeable container, like filter paper type of thing. So solvent can sort of pass through that membrane, but leaves the solid material inside. And we've got the solvent down here in this flask, and that's getting heated up to boiling point. So what happens is the solvent vapor comes up here, and if we heat it up enough, the solvent vapor is going to go all the way up to the top, and at the top there is a condenser. So it cools the solvent, so it goes back into the liquid state, and it drops down onto your solid sample here. So we have enough solvent evaporating to fill up, so the solvent sort of drips down and it fills up this container here, and it's also sort of coming up through this section here, and eventually when it gets to this level, the solvent can drop back down this tube into the original flask. So that's where we're sort of recycling the solvent. So the solvent is getting evaporated, but it condenses, and then it fills up this middle section, and eventually sort of comes back down into the bottom section. But at that point, it has interacted with the solid sample. It's dissolved some of the material that we're trying to extract, So the extracted compounds are also dropping down into the flask here, but they're less volatile. So as we're evaporating the solvent, the dissolved material, the solute, gets trapped here. So it's slowly sort of getting sort of leached out of the solid sample, mixed in with the solvent, and then sort of dropped down into the flask.
Why they are not able to evaporate or to go off the solvent?
Yeah.
Why is trapped on the bottom?
Because it's not, it has a higher boiling point.
Okay.
Effectively,
Point.
yeah.
Okay.
So it's less volatile.
Yeah.
So even if some of it does evaporate, it's a much, a small proportion of it will evaporate at any one time. So you're sort of I think usually you sort of just turn this on and leave it running for a few hours, but you don't have to top up the solvent. It just sort of keeps on evaporating, condensing, and sort of cycling through the system. And eventually you sort of dissolved enough of this material and transferred it into the flask at the bottom. So if you're doing this, without any heating and just sort of using one batch of solvent without this sort of continuous process, you would need a much larger volume to extract the same amount of material. Okay, so now get on to extraction of one liquid with another liquid, and this is where we need to, choose liquids which don't mix with each other, so that they will separate from each other during the process. So they have to be immiscible liquids, and it's usually water and some organic solvent which doesn't mix with it, like hexane or chloroform. And so usually what we're doing here, well, it could be the other way around. So, maybe we are trying to extract something which is relatively non-polar, which is in the water, and extract that into the organic solvent. But it could be that we are trying to clean up an organic solution and remove polar materials from it by washing it with an aqueous solution. And we need to, when we're sort of thinking about what's going on here, we need to think about the sort of rate, the relative solubility of the compound of interest in the two liquids. So it's going to be the ratio of the concentration of that material in the two liquids once we've allowed it to reach an equilibrium state. So, and that's referred to as the partition coefficient, sort of labeling it P here. It can sometimes be labeled as K, which kind of relates to sort of broader thermodynamic calculations. We're using P for partition coefficient here. So it's just a ratio of concentrations. In this equation here, if you're being really precise, you have to talk about something called the activity of a molecule in a solution. Does anyone? I don't know. I've never done enough sort of thermodynamics to really understand this. Does anyone here know about activities as opposed to concentrations? Good. No, I don't have to talk about it now. But I think it comes in if you've got a very concentrated solution. You have to sort of think about this more sophisticated term called the activity. I think it comes in when you've got interactions between the solute molecules. But we're only really thinking about relatively dilute solutions where we just need to think about concentration. So concentration represented by these square brackets here for solute X in two different solvents, two different liquids, 1 and 2. So the partition coefficient is just the ratio of the concentrations of the that same molecule in the two different liquids. So, a couple of different examples here of mixing water with either chloroform or hexane. So why do you think we've got the liquids, the hexane and chloroform, and different? Yeah, that's right. So chlorinated solvents are more dense than water in general. Most other organic solvents like hydrocarbons are less dense, so they sit on top.
Going back to that activity, the activity distinction, is there going to be any benefit in pushing that in this program?
No. No. Yeah, concentration is accurate enough. Yeah. Yeah. So you can have these different mixtures, and we need to think about the sort of relative concentrations of the solute X that we're trying to extract. Now, actually, at this point, I think I'll bring in just a little sort of video demonstration of this process in action. So, this partition coefficient, this is an equilibrium property. So, what we're doing is, if we've got the solute in one liquid, we're trying to extract it into the other liquid. We could just put them together and wait for the sample molecules to slowly pass across the interface between the liquids and sort of get into the other solvent and eventually reach their equilibrium state. But if we want this to be a convenient process, we need to accelerate the equilibrium state. And so what you need to do is, if I can just find this video, where's it gone? Okay, here it is. Don't think this will come out in the recording, but I'll post this on Moodle. So if you did the lab classes last term, you've done this yourself, but for those of you who haven't, this is what's happening in a solvent extraction process. So this has got a water solution here. It's got some growth media and antibiotic in it, and this is an organic solvent. It's an absolute glass state. So in order to mix them together and to accelerate the equilibrium state of the antibiotic that we're trying to extract between the two liquids, you have to increase the surface area where the liquids contact each other so that you give the molecules more opportunity to move from one liquid to the other. So you have to shake them up together, which I'm about to do. You have to really vigorously mix the liquids. It might not come through on the video, but as you're doing this, it looks like it's almost a homogeneous mixture. But what you're doing is you're just creating lots of small droplets of each liquid within the other liquid, and there's a much greater surface area of contact between them. And it takes a while for the liquids to settle out, but eventually you can see two separate layers coming back again with a sort of clear layer on top.
So that's the extraction.
Yeah, and they've now settled out, so you can just sort of drain them through the tap at the bottom of the apparatus.
What's the process again?
So this is the solvent. We're still in the solvent extraction procedure here.
The separation. So you've expanded the surface area and the actual product of the product. You're separating between the liquid and antibiotic.
Yeah. And so it's going in from aqueous solution into the organic solvent as well. So while we're doing this process and we're sort of trying to extract a molecule from one liquid into another, we're interested in how efficient this is and how much of the sample remains in the original liquid, and therefore how much of it has been transferred into the other liquid. We can think about this term Q, and this is just a measure of the fraction of the sample molecule, the solute that we're trying to extract, how much of that is left behind in the first liquid. So phase 1, that's the original liquid that it's present in. Phase 2 is going to be the solvent that we're using to extract it into. So it's just this ratio of how much is left behind after the extraction compared to how much was there at the beginning. But this needs to be quantified a little bit more in terms of the concentration of the material in each of the liquids. So it's just, what we're trying to do here is, how much mass or how many moles of the material in each of the different phases. So it's going to depend on the concentration of the solute and on the volume. So if you multiply those together, that tells you how much of it is there, either in grams or in numbers of moles. And so, the amount that's in the original liquid, phase 1, is the volume of that liquid times the concentration which is present there. And the total amount is that term there, the amount of the material in liquid 1, plus the amount of material in liquid 2, which is just the volume of the second liquid times the concentration which is present. And we can, cancel out or divide the top and bottom parts of this term with the concentration. So we just get the volume of the first liquid here above and below the bar, and this term here where we're multiplying the volume of the second liquid that we're using to extract the material over the partition coefficient. So the ratio of the two concentrations, which is the partition coefficient, and this partition coefficient is going to be a constant for a particular molecule, for a particular pair of solvents. So if we know what we're extracting, and there's some understanding of how it behaves, we already know what that partition coefficient is if it's a system that's been worked on before. So I'm bringing in this level of detail so that we can justify some of the practical aspects of doing the solvent extraction process. So what we're going to do is work through this equation to demonstrate that you can get more efficient extraction if you divide the extraction solvent into smaller portions and do repeated extractions. So in the lab class last term, we did this a little bit, not a very sophisticated version. We didn't even quantify the results. But if you remember, what we did was, each time you did an extraction, you used 2 separate portions of the extracting solvent, the ethyl acetate, and then mix them together at the end. That is more efficient than just putting all of the ethyl acetate together in one portion. So, if you think about this in molar terms, number of moles of a molecule, then if we start off with n moles, then we end up with Q times n moles at the end left behind in the original solvent. But if we then repeat the extraction with the same volumes, it's just, the second extraction, the fraction that's left behind, Q, is going to be, it's going to be the same sort of ratio involved. But the overall process from start to end, after the first extraction and the second extraction, we can work out the amount that's left behind in the original liquid by multiplying this mathematical term by itself, depending on how many times you repeat the extraction. So if you do 2 extractions with the same volume, the fraction that's left behind in the original liquid is going to be this V1 over V1 plus V2 squared. So I'll work through, before I work through the equations, I just want to sort of try and illustrate this more visually. So imagine, we've got something dissolved in solvent 1. I'm just going to illustrate this with some little dots for the molecules that are dissolved in there. And just a simple case where the partition coefficient is 1, so at equilibrium the molecules are equally soluble in both liquids. So mix them together, allow equilibrium to be achieved, and we've got the same volume of each liquid to each other. So that means you're going to have the same number of molecules in each of those 2 liquids if we have this partition coefficient of 1. So it's equally soluble in both. So that's 24 dots there, so it's going to be 12 here. So in one extraction, we've extracted half of the molecules. So now, I've left myself enough room, but I'm going to start again. So from the hand-dry tank, but using half the volume of solvent 2. Yeah. Again, 24 molecules. Mix them together. So the partition coefficient is still 1. So these little red molecules are equally soluble in the two liquids, but there's only half of liquid 2, so there's only going to be a third of the molecules end up in liquid 2. So starting off at 24, so that's going to be 8. So now going to remove that top layer. And replace the top layer with another portion of solvent 2, which has nothing in it. And we've got 16 molecules there, and we mix it again. The numbers don't quite work out, but if we, let's say, approximately less than 15, so yeah, let's say we get 5 of them into here and leave behind 11. So take out that top layer, and we've sort of kept this aside, and we put those two together, and we have now extracted 1, 2, 3, 4, 5, 6, 7, 8 plus 5, 13 of the molecules, and okay, it's not a huge difference, but it's hang on, I calculate this one. Yeah.
16.
Yeah, it's not a huge difference, but we have extracted more of the sample using these two smaller portions than we did with just one portion. And that's a general feature of this technique. If you can afford the time to do additional extractions, then it becomes more efficient.
So is this a contrast between the two, the solid phase and partition coefficient? Is this an evaluation?
Not really. It depends.
Demonstration of two different techniques.
Yeah, two different techniques, which depending on what experiments you need to do, one may be more suitable than the other. If you can't find the suitable solid phase extraction material, then you have to do solid extraction. If you can't afford the solid phase extraction material, you might have to do the solid extraction. But what I'll now do is, got a few minutes time. rather than doing a visual demonstration, just go through it and using the equations. So we take this example here where we've got a sample of water and it's contaminated with benzene. So it's going to have a relatively small concentration of benzene in it, but we're trying to extract the benzene out so that we can quantify it. And we're going to use a non-polar solvent like hexane, which will dissolve the benzene out of the water. And we need to know, it's a relatively sort of common molecules we're working with, so we can look up what the partition coefficient is for benzene between hexane and water. Now, these partition coefficient ratios, they're going to cover a very wide range of orders of magnitude. So that kind of quantity is often converted into the logarithm, and that gives you a smaller numerical range. So has anyone done any sort of medicinal chemistry studies? Had a log P for drug properties at all? Yeah, I mean, so there's a term which is used to describe the properties of drugs in terms of how they behave in the human body. So you might, if you're sort of reading about drug design, you might see people talk about the log P of a drug. It's a way of sort of taking it's sort of describing how it behaves in biological membranes compared to aqueous solution. And it's the same kind of principle. It's taking a property which can cover a wide range of numbers and sort of bringing it down into a smaller numerical scale. Also like pH. We talk about pH values in terms of this sort of quite sort of limited range from 0 to 14 or whatever. But that is representing a very, lots of orders of magnitude of proton concentration. But so it's often going to be stated as a logarithm. So we need to do the calculation, we need to convert that into the original partition coefficient. So we need to take the logarithm to get the original partition coefficient. We need to sort of go do this calculation 10 to the power of log p. So it's 2.06. If you want, you can sort of check this on a calculator, but 10 to the power of 2.06 is 114.82. And we now have that partition coefficient value that we can put into the equation. We know what volumes that we're working with, 10 milliliters of water, 30 of hexane. B1 is the water, the original solvent. B2 is the hexane we're using to extract. So 10 goes there, 30 goes there. So you put all the numbers in and get the calculation. So it turns out, this kind of log p value implies that hexane is not very soluble in water and much more soluble in hexane. Because when we do that one extraction, we have a tiny fraction of the benzene left behind in the water. But we can make it even more efficient if we divide the hexane into 3 smaller portions, 3 times 10 milliliters. So just considering in percentage terms, so it's only 0.2895% of the benzene left behind in the water, meaning that we've got 99.71% of the benzene dissolved in the hexane. But now we'll sort of go through the calculation for these repeated extractions. Same overall volume, but we've divided it into smaller portions. So each time you do the extraction, you will leave behind a certain proportion of the benzene in the water. Then when you do the next extraction, you're going to that original sort of small amount of benzene that's left behind is going to get reduced even further, and it's going to get reduced by the same factor each time you do it. So if we're going to end up with a slightly different number here because we're using a volume of 10 milliliters instead of 30. But once you sort of go through that calculation within the brackets there, each extraction that you do, the proportion that's left behind is 10 divided by 1158.2. If you do repeated extractions, the second extraction, you're going to multiply the original value of Q by the same number. So it's going to be Q squared for the second extraction. Then you do a third extraction, you multiply by the same number again. So it's going to be the original, call it Q1 for the first extraction. That value, if you do it 3 times, is going to be Q1 cubed. Gives you the overall proportion that's left behind. And that multiplies out to this 6.4 times 10 to the minus 7. So the percentage remaining is 6.4 times 10 to the minus 5, which you can basically ignore because it's so tiny that it really doesn't make any difference. So we've generated a very efficient extraction here where you've got virtually all of the benzene has been extracted by dividing it into these smaller portions. So that's, I think I've kind of run out of time for tonight. So I'm going to come back to this at the beginning of the next session just to discuss what happens if you've got ionized material or compounds which are in an equilibrium between neutral and ionized states, and how we get around that by altering the pH of the solution. So that's just a few more slides on that, and then we're on to the chromatography.